At this point, we're going to go ahead and move on in our textbook to Chapter 5, which explores the structure of our cellular membranes. So in this short video, we're going to talk about membrane structure. What are the components that comprise the cellular membranes? And then we will talk about how membrane fluidity is influenced by some of the different components that we find within the cell membrane. When we think about the biological membranes, we automatically think of plasma membrane, the membrane that's going to differentiate the inside of the cell from the outside of the cell. But there's membranes all throughout the cell, things such as the endoplasmic reticulum in the Golgi apparatus. The nucleus is encompassed in a membrane as well as multiple organelles. Even vesicles that are released from the Golgi and make their way to the plasma membrane, those are made up of membranes. So when we're talking about the membranes within a cell, we want you to think not just about the plasma membrane, but about all the membranes. And what are the functions of those membranes? Well, there's several key functions, including things like compartmentalization. We just talked about cellular respiration, and most of those reactions that we talked about were happening in the mitochondria. And the fact that you can localize key enzymes into a specified compartment is absolutely critical to a lot of our cellular functions. Selective permeability is incredibly important, determining what is allowed to enter into the cell or into a particular compartment, and what is extruded out from that compartment. For instance, waste products that the cell produces and wants to release out into the extracellular space. That's all mediated through the various membranes. In regards to scaffolding our biochemical reactions, what the membranes allow us to do is to stabilize the position of those reactants and localize those in relative proximity to the appropriate enzymes to drive particular chemical reactions, kind of allowing the appropriate reactants and the appropriate enzymes to be localized together in the same region. Cell signaling also is largely due to what is transpiring at the various membranes, regulating what signals are received from outside the cell in versus what is happening within the cell, that those signals can get transmitted out. It's also where we're going to be initiating our signaling transduction cascades within the cell, and we also can have cell-cell communication at the cellular membranes between cells as well. The framework of the membrane is really the phospholipid bilayer. The phospholipids, just to remind you, are amphipathic molecules. They have the hydrophobic region, which is comprised of those fatty acid tails that is going to face towards other fatty acid tails. And then we're going to have a hydrophilic head group, which is going to face the aqueous environment within the cell and the extracellular space that is also aqueous. So now I'd like to explore some of the key components that make up our cellular membranes in addition to the phospholipids, which we've already talked about. As we're looking at this image, you can immediately see that we've got large proteins that traverse their way through the phospholipid bilayer, and these are known as integral proteins. In addition to the various types of proteins, which I'll talk more about in a minute, we have things such as cholesterol, which also help to make up the phospholipid bilayer. And cholesterol is absolutely essential in terms of maintaining membrane fluidity within the cell, and we will talk about that more as well. One thing you'll notice on cholesterol is it has this hydroxyl group, which is going to interact with the extracellular space or the intracellular space, but the bulk of the molecule is going to be nested there with the fatty acid tails in the phospholipid bilayer, because this is all nonpolar. In addition to proteins and cholesterol, we're also going to find things such as glycolipids and glycoproteins. So glyco, again, would imply a sugar, and so what you can see here is we've got a protein with a series of sugars that are extended out into the extracellular space. And this is known as a glycoprotein. And the glycoproteins, as well as the glycolipids, they can act as signals for the cell a way to identify a particular type of cell. Oftentimes they're going to be involved in maintaining an appropriate immunological response. So these are oftentimes you'll see just involved in signaling. So again, what we have that make up our membranes are going to be proteins, cholesterol, the phospholipids, glycoproteins, and glycolipids. The other thing to point out is that the phospholipid bilayer, we can talk about the two halves of that we refer to as the leaflets. So one leaflet, the one that's interacting with the extracellular space, the one depicted in this image on top, could be different or asymmetrical to the leaflet that we find on the intracellular space. Before we move on, I'd like for you to take a minute and to think about why is it that we refer to the membrane as the fluid mosaic model? Why do we think it's fluid, and what component of it is actually a mosaic? So if you could take a second and think about that before we move on, that would be great. So when we say the word fluid, what that implies is that those phospholipids are constantly in motion. They are moving around throughout the phospholipid bilayer, and they're not stagnant and fixed in one place. I like to think of them as kind of waves on the ocean, but actually they are extremely dynamic and actively moving in that space. And it's not just the phospholipids. The cholesterol can migrate through. The proteins are moving around. Most of the proteins are moving around. And so we think of this in a 2D, very fixed state, but actually it's an active, dynamic structure that is constantly in flux. Now, why do we say mosaic? Again, the phospholipid bilayer is not just made up of phospholipids, but as we discussed, it's made up of proteins and cholesterol and these various sugars that are attached to lipids and attached to proteins, etc. So it's really a mosaic of different components that comprise this whole structure. All right, so far what we've covered is membrane structure includes phospholipids, cholesterol, proteins, and various carbohydrates that are attached to those lipids and proteins. So now let's talk a little bit about the proteins that we find in the cellular membranes. So I want you to go ahead and take a second to answer the following question. Which of the following is not a type of membrane protein? Go ahead and answer that. Hopefully you chose answer B. The nuclear receptor proteins are not found in the cell membrane. They are, as their name implies, found in the nucleus and actually act as transcription factors. Even though we haven't talked about that in our class, the name implies that they do not reside in the cell membrane, but rather in the nucleus. So let's talk a little bit about the membrane proteins that we're going to find nested in the phospholipid bilayer. So the first one I want to talk about are these transmembrane proteins, also known as integral proteins, and these cannot be released from the membrane. So the transmembrane proteins are physically inserted into the hydrophobic interior of the phospholipid bilayer. If you think about the structure, the nonpolar amino acids that are able to interact favorably with the hydrophobic fatty acid tails of the phospholipid bilayer. The other integral protein would be the lipid-anchored proteins. And so what we find here is that the lipid molecule is covalently attached to the protein itself, and it keeps it anchored there. So the last protein that I wanted to discuss are the peripheral membrane proteins, and these are non-covalently bound to regions of the integral proteins, and they project out from the membrane. So again, the three types of proteins you should be familiar with are the integral proteins, which include the transmembrane proteins, the lipid-anchored proteins, and then we've got our peripheral membrane proteins, which have an interaction here with the transmembrane proteins, but are not physically and covalently attached. Let's briefly discuss what some of the key functions of those membrane-associated proteins actually are. Some of those functions include things such as transport. So things like the transmembrane proteins are going to be able to facilitate the import and export of important solutes and various ions, chemicals, etc. into and out of the cell. Some of those proteins also have enzymatic activity that can begin certain catalytic reactions. They also can be important in initiating signal transduction cascades. So as they're nested there in the plasma membrane, they might receive a signal from the extracellular space and transmit the message of that signal to the interior of the cell. Those proteins can also be involved in cell-cell interaction, either responding to another cell, transmitting a signal to another cell, for instance. And then lastly, some of those transmembrane proteins are important in attaching to the extracellular matrix, which provides some stability as well and keeps the overall cell somewhat in a more anchored position. All right, so let's do another assessment question. Which type of molecule can be attached to membrane lipids or proteins to aid in its recognition by other cells? Go ahead and take a minute to answer this question. The correct answer here is A, polysaccharide. Earlier on, we had discussed the idea of glycolipids or glycoproteins. Both of those have various number of sugars or carbohydrates that are attached to either the lipid or the protein that's nested in the phospholipid bilayer. So in summary, for membrane structure, you should have a solid understanding of what exactly are phospholipids and their general chemical properties, as well as cholesterol and how it's nested there within the phospholipid bilayer. We've talked about various proteins such as transmembrane and lipid-anchored proteins, which are our integral proteins, and the peripheral proteins, which are non-covalently interacting with those transmembrane proteins. We also mentioned glycoproteins as well as glycolipids, which often are involved in cell recognition and cell signaling. Right, so we have thoroughly discussed membrane structure, and now I want to briefly explore the idea of the fluidity of membranes and how that can be modulated or modified. As we've already discussed with regards to the fluid mosaic model, the phospholipid bilayer is constantly moving, and those phospholipids are moving rapidly. So I want you to take a second and answer the following question. How quickly do you think those phospholipids are actually moving? And I want you to rate the following 4 different possibilities in terms of how rapid you anticipate that movement actually is. So rate them going from 1 to 4, with 1 being the fastest and 4 being the slowest. Go ahead and take a second and put them in order. The fastest type of movement for phospholipids are their rotational movement. So the fact that they are rotating around an axis. The second fastest movement within the cell membrane is the lateral movement of those phospholipids. They're moving all throughout their particular layer. The third fastest movement would be you actually going and getting a burger or, say, a burrito for lunch one day. What happens quite slowly is the movement of a phospholipid from one side of the phospholipid bilayer, from one leaflet to another. So here's showing some of those movements. Again, the rotational movement, that movement around an axis, is happening extremely quickly. Movement throughout the phospholipid bilayer and migrating throughout that leaflet is happening at a very rapid speed as well. What happens extremely slowly, just about once every day or so, is a phospholipid moving from one leaflet into the other leaflet, say from the leaflet that's interacting with the extracellular environment and moving all the way through that phospholipid bilayer to the intracellular environment. And if you think about the structure of a phospholipid, that polar head group migrating through the fatty acid tails of the neighboring phospholipids is energetically very unfavorable. It's so unfavorable that it actually requires ATP to be put into the system. So this is not going to be happening very rapidly. Each lipid is said to be moving at approximately 36 miles per hour. So if this phospholipid was the size of a car, this would be equivalent to traveling 684 million miles per hour, which is beyond our comprehension. So again, we tend to think of the phospholipid bilayer as this stagnant 2D image, and really, as I mentioned earlier, it's incredibly dynamic. And why is it that we actually want the phospholipid to be fluid? Well, we want some partial fluidity because if it was completely stagnant, then that membrane could potentially rupture and break open. We need mobility within the membrane so that we can do things like transport things into the cell through the phospholipids, or vesicles can migrate throughout the cell to be exported out. And we'll talk a little bit more about this later on in the semester. But here what you can see in this image is we have a vesicle within the cell, and it's going to be able to fuse and merge with the existing phospholipid bilayer such that the phospholipids that comprise that vesicle are now a part of the cell membrane. And what had been inside the vesicle is now extruded out of the cell and dumped into the extracellular space. So this is a great way to bring things in or to send things out. It can't be completely fluid because we do need some structural integrity. We don't want to just let anything into the cell. So the fact that the phospholipid bilayer is partially fluid is absolutely essential. We want to be able to bring things in and send things out, but we don't want it so leaky that anything can enter, and we don't want it so rigid that it's going to break. All right, so let's talk a little bit more about some of the things that can impact the fluidity of the membrane. The first thing I want to talk about that affects membrane fluidity is temperature. As we start to warm up a cellular membrane, it's going to become more fluid. Whereas if a cell were to find itself in a colder environment, it's going to be less fluid. And you know this from working in the kitchen. If you were to take something like butter and put it into a warm pan, eventually that becomes liquid, right? Or even coconut oil, for instance. So increase in temperature is going to make that phospholipid bilayer more fluid, and putting it in a colder environment is going to make it more rigid and less fluid. Secondarily, the composition of the fatty acid tails is actually going to matter. So I want you to take a second and answer the following question. Which do you think would be more fluid, saturated or unsaturated fats? Now again, thinking about what you know from working in the kitchen, think about a saturated fat such as butter versus an unsaturated fat such as some of our plant-based oils like an olive oil. And we know that the unsaturated fats are much more fluid, and the same is true here. So if we have fatty acid tails that are part of the phospholipids, they're going to be less tightly packed just because that double bond is going to create a kink in the structure. As opposed to a saturated fat, those fatty acid tails are going to be able to line up more tightly and closely to each other and give it more structural integrity. Third factor affecting membrane fluidity would be the length of the fatty acid tails. For shorter tails, there's just less to interact with one another, so those are going to be more fluid. So as the length of the fatty acid tail gets longer, you're going to become less fluid. And then lastly, I want to mention cholesterol. Because of the ring-like structure on cholesterol, it is known to be less prone to the temperature influences when you compare that to the fatty acid tails found in the phospholipids. So at really high temperatures, cholesterol is able to make the plasma membrane less fluid, whereas at really low temperatures, cholesterol is able to make the membrane more fluid and prevent freezing. So cholesterol really is incredibly important. For most cells, it makes up somewhere around 20% of the components in the cell membrane, but you can find certain types of cells where the amount of cholesterol is as high as 50% of the components in the plasma membrane. All right, so just to dive in a little bit deeper on each of those things that we discussed. So again, the composition of the fatty acid tails when we're looking at saturated versus unsaturated fats, we said that unsaturated fats, when we increase the number of double bonds, that's going to increase your fluidity. Again, if we have a bunch of chains of saturated fats, they're going to be able to interact really nicely with one another and provide much more stable structure. Again, the fatty acid tail length, the shorter the tails, the more fluid it's going to be. So if you can increase the length of your fatty acid tail and make it longer, there's just more fatty acids to interact with, and that's going to decrease the fluidity. And lastly, as I mentioned, cholesterol, because of that ring-like structure, this is just predominantly all carbons and hydrogens with the exception of that one hydroxyl group that we mentioned earlier. This is not as strongly impacted by changes in temperature. And because of that, it's got a somewhat rigid structure. And as I mentioned, at really cold temperatures, this almost acts like an antifreeze and inhibits freezing of the plasma membrane. Whereas at really high temperatures, when the phospholipids would get more leaky, cholesterol provides some structural integrity there as well. So cholesterol really is incredibly important for our plasma membranes. All right, so this just summarizes everything we just talked about as it pertains to membrane fluidity. The length of the fatty acid tails is incredibly important. The presence of double bonds due to our unsaturated fatty acids is going to affect our fluidity. And lastly, the presence of cholesterol is incredibly important to maintaining the fluidity of the membranes. All right, so let's take a second and answer the following question. If a cell's membrane was becoming too solid, what could the cell do to compensate for this and to maintain its normal level of membrane fluidity? Answer B was the correct answer here. Increase the number of unsaturated fatty acid tails. Let's think about the other answers and then come back to B at the end. So heating up the membrane would certainly increase membrane fluidity. However, we are not able to just heat up our membranes, and the membranes are going to become leaky if you heat them up too much. So even though theoretically A makes sense, physiologically it does not make sense. What's wrong with answer C? Increase the number of saturated fatty acid tails. Well, again, saturated fats are going to be able to line up, and they're going to make the whole phospholipid more rigid. So that's going to decrease fluidity, not increase fluidity. If we make the fatty acid tails longer, does the same thing. You're going to be decreasing fluidity because, again, you've got more fatty acids to interact with. So the correct answer, again, is answer B. If we increase the number of unsaturated fatty acid tails, you're going to be creating those double bonds and a kink in the structure, and that's going to result in an increased fluidity in the membrane. And actually, it's incredible, but our cells are able to adapt to changes in temperature by altering the lipid composition of their membranes. And one of the ways that we are able to do this is through desaturases. So there's a whole family of enzymes, such as steryl-CoA desaturase, and these are able to introduce a double bond in the fatty acid tails of our phospholipids so that indeed the membrane can become more fluid. So as the temperature gets colder and our membranes decrease in fluidity, we can induce the expression of certain genes such as the desaturases, and that's going to cause certain fatty acid tails to become desaturated, and that will make our membranes more fluid even though we are in a cold environment. The last thing I want to mention is that even though I've talked for the last half hour about membrane fluidity, I don't want you to get the idea that everything is fluid. So there are certain proteins that can be attached either to the cytoskeleton, that's a skeleton inside the cell, or the extracellular matrix, which is on the outside of the cell, and sometimes we'll find that certain transmembrane proteins can be fixed in place to either the cytoskeleton or the extracellular matrix, and this just keeps some of those proteins in a more fixed and locked position such that not everything is actually moving around all the time in the phospholipid bilayer. All right, so that completes our discussion of both membrane structure as well as the fluidity of membranes. Please bring your questions to class and we're happy to help you.